good to have you all here. So I want to talk about a bit, of course, about sustainability and specifically energy and CO2 consumption, let me say, or CO2 emission in this case. And I've been doing this for a few years now, or for a longer time, most at Merck. Yeah, and I've seen a lot. So we are on a journey here through all the utilities, but also HVAC systems, and I want to take you on this journey now for the next hour to talk a bit what we see in practice. So the Merck healthcare business has more or less 20 manufacturing sites around the globe. We make biotech APIs, finished products, tablets, chemical APIs. So we have a variety of sites in here, and it is very interesting to see where you can find the best gains, what are the best ideas in practice to reduce energy consumption, and there's still a lot to do. So we have a lot of work to all the engineers here, yeah, and also the quality people and operations. There's still a lot to do. First, let me start with this nice figure here. So if you look at these companies, I also put some tech or IT companies, let me say, or data companies in this here, as well as Endres, yeah, or Grundfos, not only pharma companies, but I thought, okay, when are these companies going to be CO2 neutral? I looked it up because everybody has got a sustainability report and you can clearly look it up. It's on all the web pages. And so we have a diverse picture here. There are some companies like Merck Inc. Yeah, this is not where I work, or MSD in other countries than the US, who already want to be CO2 neutral. I don't know if they made it, yeah. But it's a tough way. I would say it's not easy. Yeah? You need a lot of measures. You need to reduce. You need not only just install heat pumps somewhere. Yeah, it's not the only way. It's much more difficult, and you also need the capex, the money to invest for this. Yeah, so. And it's quite interesting. So yeah, many sites around 2030. Let's see if they're gonna make it, but pretty interesting to see. In Germany specifically, I can say that 2040 is the latest option. Yeah, so this has been set up by the government that 2040 shall be the end. Good. And on this journey, I have seen that if you really want to achieve CO2 neutrality in pharma manufacturing, there's only one way, which has well been described by CRB, yeah, in a good article. So the first thing, and I still work a lot on this, is reducing energy consumption. Yeah. For all the finished product sites, of course, HVAC is the first thing you should do, because there are the biggest savings in it, and also not too much invest necessary. So here the business case is excellent. Yeah. But also the utilities, store water cold instead of hot, or generating it in a cold way, membrane-based technologies. Optimize cleaning recipes. Yeah. So why do you need a hot final rinse? Well, then people always say the drying process is much quicker than if you have a cold rinse in equipment. So we see lots of discussions, and this is what I want to tell about here now. And then you got to recover the rest of the heat that is left. Reuse it where you can, and the same also applies for water consumption. Yeah, not only heat recovery but also water recovery is a nice topic. This is in Europe the hot topic we're discussing now. In the US, the USP just has emitted or updated the Q&A question on this. Can you reuse pure water for pharmaceutical purposes? Yeah. Yeah. So, and then you start using the heat pumps, and there's more or less two types. First is, let me say, the standard heat pumps that go up to 65, 70 degrees. And then there's the high temperature heat pumps. This technology is, still quite new in our industry, I would say. Of course, here the efficiency, the so-called coefficient, the COP, the coefficient of performance is not as high as for the standard heat pumps. Yeah, but this is where you can go to higher temperatures. For example, when you want to sterilize, you need steam of, let me say, 135 degrees or so, and then you need higher temperatures. Yeah? And then the last of these 4 steps is the electric boiler. Yeah. In Europe, we've already seen first sites that have no gas or oil connection anymore. That is wonderful. Merck is not under them. So we have one site in Uruguay that has made it already. Yeah. But that was not too complicated, let me say. Anyway. And now this is also very interesting. This can give you some guidance if you also work in a global environment or are at a machine supplier, equipment supplier. So I divide the pharma manufacturing in 4 types of manufacturing sites. Yeah? It is chemical API and then finished product. So this means tablets, OSD manufacturing, tablets, capsules, stuff like that. This is very popular. And then you have the biotech APIs and then the fill and finish sites, the sterile fill and finish sites. This is more or less how I divide it. Of course, there's also manufacturers of ointments or non-sterile liquids or stuff like that. I would say this belongs then here into the OSD part, more or less, for me. And if you look at these sites, I've put here a table that shows where you should start to look at, if not yet done, where the biggest energy consumers can be found. And so in most cases, as you see, it's HVAC. HVAC systems account typically here for more than 50% of the entire energy that is being used, and you can reduce a lot. Yeah, I'm gonna show you afterwards some examples. There's so much you can do. For example, in a tablet facility, you don't need 20 air changes like it was written 20 years ago in the aseptic guide FDA. They were writing 20 is sufficient typically. Yeah, it is sufficient, but 8 is also maybe sufficient, 8 air changes per hour. Yeah. And this is a huge saving, I can tell you. Yeah. Warehouses, bring down your warehouses. Yeah. Just base it, so especially for the Asian countries or Southeast Asia, where it's quite hot. Typically, you only have to cool most of the day. Yeah. So, and then base the air change on the cooling load. Yeah, just use as much airflow as you need to cool the building down. That's it. Yeah, as an example. Good. HVAC, the only facility type where a different process needs more energy. This is the chemical API sites, really, where you have chemical reactions, where you have crystallizers, where you have centrifuges to dry the API later, and so on. So this is typically where the process itself consumes most of the energy because you are liquefying the raw materials, and this consumes a lot of energy. And typically, this type of manufacturing happens in CNC areas, so they only have very little non-sterile clean rooms. So here it's the process. Yeah. Then if you look further, cleaning and water systems. So why do I count these together? Because if you look in practice, if a water for injection system is making hot water that is consumed mostly by the washers, by whatever it is, pellet washers, equipment parts washers, SIP systems, whatever, then it doesn't matter if you account the energy emission or the carbon dioxide emission to cleaning or to water systems. It's connected, just think as a facility. Don't think in departments, think as a facility. You are one facility, one team. Try to get it down together. So compressed air, don't underestimate compressed air. And we've done a lot here in the past. I'm doing a global program at Merck on compressed air. And the savings are not as big as in HVAC, but we see substantial We see substantial savings here as well. And I can tell you, we've updated an ISPE guide 2 years ago, rewriting specifications. Yeah. So, because that was always a problem in the practice, how can you set a proper specification for compressed air and gases? There is this nice ISO standard 8573, which is pretty common, but this only defines classes. It doesn't tell you as a pharma company what the right class is for you. So you can't basically use it. You can use classes, but need to define your own spec risk-based. And that's what why we've updated also the ISPE guide on gases to give the people some info, some guidance, what makes sense. And I'm going to show some nice examples later. Good. Yeah. Last but not least, we talk about the process equipments. Yeah. So like in OSD, the granulators, like tablet presses, packaging lines, and so on and so forth. Yeah. So every type of facility has its own equipments. Yeah. In the steroids, it's the filling lines, it's the compounding vessels, it's whatever. Yeah. But this is typically the order where you should look at energy consumption. Good. And HVAC. So I'm going to go through all these types of equipments over the hour now. Yeah. And give a bit of experience where it's really useful to look and what to do and how it goes. Because really at Merck, we've looked at all types of equipments now. And yeah, so with HVAC, of course, lower air changes. This is the easy, the quickest part that brings a super saving. And I'm not only talking about production times, but also non-production times. If you have on weekend no shifts or you have a maintenance shutdown, then you can even further bring down the air changes. Yeah, recirculate. Why do you need so much fresh air? Yeah, 90% recirculation is no problem if you have proper HEPA filtration. Yeah, differential pressures, they bring a little less energy savings, but nevertheless, you can have a look at it. Filters, do you really need H14 HEPA filters that have even 1 log higher reduction, separation efficiency than the H13 filters? And to be honest, if you have, let me say, a non-sterile clean room, you could even achieve the clean room specification with an F9 filter. Yeah, HEPA would not, basically not even need it, but then you have the cross-contamination topic. So here is maybe still advisable to have these HEPA 13 filters, but check it out. Humidity limit also brings a lot. Yeah, so you can turn down really or look at the specification. That is very, very interesting. Do you really need a humidity limit? Yeah. Let me imagine you have a warehouse and you store hermetically closed product in drums that are really closed, where no air can reach inside, can go inside. Then temperature still has an impact. So you need to control temperature in the warehouse, but humidity does not. So you could say in the warehouse, if all product is closed, you don't have a humidity spec and don't need to humidify or dehumidify as necessary. Yeah. So, and this saves a lot. Then adiabatic humidification of the exhaust air is a topic. So there's numerous ideas. Yeah. Or one other topic, I've said this already in the warehouse, reduce the air changes just to meet the cooling loads in summer, yeah, or when it's hot. Yeah, in Southeast Asia, it's always hot. Okay. So, and I can tell that we've already saved in our sites 1.3 million cubic meters per hour of airflow. Yeah. So this is a mid-sized sterile facility that we've shut down basically. Yeah. To compare it. And one more topic that is very interesting. So you got to understand the physics here. If you reduce airflows by half, then you need 87% less power per calculation. In reality, we see it's not 87, it's a bit less because there are pressure losses and you have old equipment and this and that. But the savings are higher than 50%. And in addition, heating and cooling and humidifying that you have done in advance is saved in addition. It comes on top. Yeah. So there is really a lot that that a lot of savings in it and a lot you can do. So we are still we started 5, 6, 7 years ago, but we still invest time and money into HVAC topics because it's really worth. We still can bring down air changes further. So from the quality perspective in HVAC system, the clean room spec is the driver. It is not something else. It is not an aseptic guide or whatever compliance document. I've looked up all worldwide, all compliance documents. Mexico is quite interesting here, but all in all these documents give recommendations, not a hard requirement, a must requirement. It is a recommendation everywhere, and we can even meet D or D non-sterile with 6, maybe in new sites, even with 5 air changes per hour. Yeah, it's possible and it works. You could even, there's also systems that vary the air change based on the particle load in the room that is even existing. Yeah, so you could even go lower, or also during night shifts or when there's no production on nights, as I said, go even lower and so on. Good. And my suggestion is when you qualify the new air change rate, leave the target a bit open, make some studies, try out the recovery time or cleanup time as it's written in Annex 1 and look where you land. Yeah. And so then you don't need to start again qualification if you don't meet the number 6 or whatever. Yeah. Make the target open and see where you land. Older HVAC systems need somewhat higher air change rates. They have higher losses, pressure losses, more leakages, and also the air flow dampers can have a technical limitation here. You can just not switch them as much down as you like. Good. Perform a pilot in the first critical clean room wherever you like. Yeah. And then apply the same concept. Learn from it and then apply it throughout the entire facility. This is how we have made really good progress, and that worked for us. Good. So that is it about HVAC for now. Let's talk about water systems. Yeah, I can say I have quite some experience in water systems and also currently chair the steering committee on utilities. Yeah. Since 2 years now, and I've assessed a lot CO2 emissions in water systems, also together with Robin, who's in the call today. Yeah, he's also doing such stuff. And if you look here, then it's clearly the steam in water systems that is needed to heat up to 70, 80 degrees, or even to vaporize in distillation units your water. This makes up 95% of the consumption or energy or CO2 emissions. Yeah. So this is really a lot. I've done an example here. I did my calculations and tried to estimate the CO2 footprint of a water system here with 3 cubic meters. This was a membrane system, but then after the cold generation, we were heating up to 80 degrees in that case. And even keeping the loop at 70 degrees instead of 80 already saves 30 tons of carbon, or 39 tons of carbon dioxide even here each year. Yeah, just switching down the temperature by 10 degrees Celsius, that is already a huge saving, and the bacteria do not survive for more than a second or some seconds at 70 degrees. If you reach 70 degrees in your loop everywhere, nothing is surviving. Yeah. So, and here on the right side, I estimated the same system, but with an ozonated cold loop. So here I have still kept some steam consumption for sanitizing the purification unit, which can't withstand ozone. So there still heat is needed. And then you see, you bring down the CO2 consumption by 90% more, even more. Yeah. So, and this is what we talk about. Still in practice, I see people hesitating to store WFI cold. And here are most of the reasons why people complain to do this and my answers to it. Yeah. So for you, if you are in the water business and people come to you, I still want to store my WFI hot, then here is answers. Yeah. The first, what they say is, We have done a cleaning validation in cleaning, and the final rinse is hot. We don't want to revalidate. What you can do then is calculate the business case, how much you save in energy, in cost, in CO2. Yeah. You go to the management and tell them about the business case. This is what makes them work. Yeah. This is the easy part. Or what you also could do is build a separate tank for the washing system. So you supply the water cold in your loop. And this one washer that really needs hot water can have its own tank and heat up on its own. Yeah, it's no problem. Next one, with a cold rinse, the drying process after washing takes much longer. This is the toughest argument. Yeah, and they are a bit right. So really, it is correct that if you rinse cold, drying takes much longer. But what could you do? You could use a fan afterwards with hot air, yeah, to dry it, like you dry your hair. Same procedure, yeah. Or I mean, typically you also see if you talk about compounding vessels or something, um, after drying, they are standing around for an hour or two anyway. So yeah, so then why not let them dry? Yeah. So really, in practice, do you really immediately need the vessel after the cleaning has happened? Do you really need the 2 minutes drying or 5 minutes drying process instead of 1 hour? Yeah. I would bet that the vessel is standing there for half a day. So, okay, sorry. Next one, but then we need to sanitize the rinse tank regularly. So if they have in the SIP unit a rinse tank and the water is not hot, then they need to sanitize. But their qualification and cleaning validation will show the suitability of your model. Yeah, it could happen that you maybe need to sanitize it. But if you have an ozonated cold loop, you could also fill up the tank patch sometimes with ozonated water. It's no problem. Yeah, it's also possible. Last one, but what about residual ozone and possible impact on API? This is also a tough one. This is a very frequent question. So the topic is, if we have an ozonated cold loop, we measure the residual ozone after the UV. And the best measurement technology and calibration methods we see on the market goes down to 5 ppb in practice. In the lab, in theory, under best conditions, you can have a better accuracy, but not in routine. Yeah, we've checked it out. And the question is, if you have 4 ppb of ozone in it and you can't measure it, does that have an impact on your API? Does that degrade your molecule? Yeah, and if you really see a risk, then make a study, conduct a study. It's possible to conduct this study just virtually in a computer, but of course the better one is live. Yeah, and then why not apply some ozone to your API in a study and see what happens? Yeah, choose the worst case API that you have. If you have several ones, look it up, try it out, and then you're good. Yeah. But yeah, people are hesitating to do such studies. Sometimes I see you can also ask external companies to do that study for you. And last hint I can give you, it's like with isolators. You know, in isolators, you're doing nothing else. There, it's hydrogen peroxide that is typically used for decontamination. But those guys are also doing a study on residual peroxide. It's nothing else. Just do the same for WFI. It's no rocket science. Okay. Good. Last slide about water. And I got to look a bit on the clock. Yeah, I have it in mind here. So hot or cold? Yeah, I mean the generation units are not really switching over. Since 25, we are really seeing an increase in the membrane-based units to make WFI, because the last pharmacopoeia that still wanted distillation units was from China, and this was changed in 25. So now there is no country anymore requiring distillation. Yeah? We have got worldwide harmonization here, which is super. And the purification units are now mostly cold. For very large systems, still vapor compression is sometimes common, what I see. But the big topic is still the loop temperature. Yeah. are you willing to go down from 70 to 20? Yeah, or whatever it is ambient. Yeah. And yeah, this was what I'm talking about, what I was talking about. So, yeah, this is the study I just mentioned. Is the API affected? This is what you should do if you want to conduct a study. And one more option I forgot. You can also look at the outlets you have. So typically there's only 1-2 outlets in your facility that really have direct contact with the API. You know, if it's a biotech API facility, let me say you are fermenting some API, yeah, then you have lots of tanks, buffer prep, cleaning, media prep, and so on. All these tanks, there's no API in it. And so 5 ppb of ozone do nothing from a risk perspective. Yeah? The only outlet that is really of concern is the one is where you do the compounding or where you have really product in it. Yeah. And you could basically put these outlets directly after the purification unit for WFI before the WFI enters the storage tank. Then you supply the 1 or 2 outlets where really you have API contact. Bring them there because the purification unit is still typically hot sanitized as it is not withstanding ozone. And then you supply all others with ozonated water and can easily have a cold loop and don't need a study. Yeah, there's plenty of options to deal with this problem. Yeah, or you have temporary heat sanitization, also a possibility. Yeah, this is even much better than a hot storage from a CO2 perspective. Good. So much about water. I've not seen any questions in the chat right now. Feel free to place questions. We're going to talk about them later, or if necessary, also immediately. Yeah. However, but then I'm going to continue talking about cleaning now. Yeah. So we've been talking about a lot about water, but also cleaning recipes. So many of you know the Sinner's Circle, yeah, which says every cleaning process is a result of 4 factors. So sorry for the German here. So it's mechanics, meaning pressure or kinetic energy, velocity, you know, where water is being sprayed on a surface. It's time, it's temperature, and it's chemistry. So these 4 factors are always play a role. And the question I thought by myself, okay, how can we get more sustainable here, and what can we do to consume less energy and also less water here? And then I came to the question, okay, what is my dishwasher doing at home? You know, I got a Miele dishwasher in my kitchen, and it's got an eco mode, you know, and I thought, okay, what is this eco mode doing differently? And in fact, it takes 4 hours for the process instead of 2 hours. Yeah. So this means it is increasing time, and that makes sense. Yeah. So if you increase time also in the pharmaceutical cleaning, you can use especially less temperature, lower temperatures, maybe also a bit less mechanics. About the chemistry, it depends. This could also happen that you sometimes add a little more chemistry, yeah, detergents, whatever it is, pH, sour or acidic or caustic, whatever it is. Yeah, and the last thing that is not in the Sinner Circle is the water consumption, yeah, that we want to reduce here. But this is for me the way to go. Yeah, go down to the cold rinse, and I've been talking about this enough, I think now. Good. Yeah, one more topic that is quite interesting. That's something we have done in a new building. So typically, when you perform a final rinse of a vessel, as an example, you have a once-through final rinse. So that means you put 200 liters of purified water or water rejection into the vessel and just let it drain, and it goes to the drain. And so my options for you is, first thing is, you can recirculate this. You can bring these 200 liters maybe to 30 liters, 50 liters, whatever it is, and let them recirculate. But therefore, it's necessary that you move away from TOC or conductivity as criteria for your cleaning validation. This is something many people have done. So they say, I rinse until I have 1.1 microsiemens like the water had before, but then you lose a lot of water. Yeah. And what we've done is we stepped away from this criteria, TOC conductivity, but we used time or flow as criteria, and then we recirculate, because if you recirculate the water, you don't get down to the conductivity you need. Yeah, you can't meet the spec anymore, but you don't need to. It's okay. Use other criteria, then you can save 90% of the final rinse. Firstly, yeah. Second idea, reuse this rinse that you have for the first cleaning step. Yeah, you have 3 cleaning steps. The first one is you clean with a potable water or softened water and you use detergent. Then you have the softened water without detergent to flush, initial flush, and then you have the final rinse. And the final rinse can also be reused for the first step because there's API in it anyway. Yeah, this is no problem, as an example. And as I said, USP has built up an opinion here, which is on the Q&A page of USP right now. Yeah, EU is also working on it more or less, but here it's more difficult. Europeans love regulations, you know, so especially Germans, maybe, don't know. Good. Yeah, there's a lot of questions incoming. Let me very quickly have a check if it makes sense to discuss now. Yeah, so would having less than 5 ppb of ozonated water to enter buffer systems be allowed? I thought within USP there's a rule, no added substances. Yeah, very good question. And you can find the answer also in the USP FAQ. Yeah, this is exactly treated there, the question. So they say it is no added substances that aren't sufficiently removed, is what they say there. So you can add ozone as long as you remove it. And we went, as ISPE, we went to USP and wanted a number. How much do we need to measure until you say it's gone? Yeah, but they didn't give us a number. They said, if you can't measure it, it's gone. Okay, so that also depends, of course, on the LOD of your ozone sensor, yeah, which may vary, but that's what they said. Okay. Next question, circularity ZLD projects. I'm going to skip this for later. Yeah, I'm not forgetting it, but we have a Q&A session later. But this question here is directly related to the water stuff we have here. Next one, how do you determine how many times we can reuse WFI? Yeah, of course, you have your quality parameters, yeah, that you measure during the purification first, if you reuse it and bring it before RO again, or if you bring it into the cleaning cycle, then I think you're gonna rinse this cleaning water anyway afterwards. So then it's a one-time cycle, let me say. Yeah, good. I'm coming back to the third question from Sanjay later, if you don't mind. Okay. Last but not least here, what I didn't talk about yet is you can also reclaim detergent. So if you are a biotech site, as an example, and use a lot of NaOH, yeah, we've seen facilities in Europe that are reclaiming the sodium hydroxide and have a lot less consumption in the entire facility of NaOH. Yeah? You need some studies, therefore. It's not so easy, but for big sites, it can pay off. Yeah, good. Sterilization. This is, I would say, the topic where the people were hesitating most, still to go down to save energy. Yeah, people are unsure and don't want to take the risk, maybe. But what we see in autoclaves and also in sterilization tunnels is that there's a lot of overkill. So, what does overkill? Typically, the basic requirement in autoclave is that you need to apply steam, saturated steam, for 15 minutes at minimum 121 degrees. Celsius. This is the basic requirement. But what we do typically is we apply a lot more heat, a lot more enthalpy on the sterilizing goods than we need. And this 15 at 121 already includes some overkill. Yeah. So, but people do a lot more. And also, if you use dry heat sterilization, it's the same. Yeah. There's the so-called F values, the lethality values that are being calculated, and they are far beyond. Yeah. So my question is, why do we need such a high degree of overkill? Yeah. Of course, nobody wants to take a risk of probable non-sterility. Yeah, it's clear. But I see that we go way beyond, and there is, just to tell you, if you want to tackle this topic and save some steam here or heat energy, there is a European Pharmacopoeia chapter 5.1.1 talking about a half cycle validation. So normally during sterilization validation, you apply a so-called full cycle, 15 at 121, 15 minutes. And then afterwards, all bacteria, so you have put bioindicators in it, 1 million per bioindicator, 1 million bacteria on a strip, or 2 million, something like that, and they should all be dead. But in this chapter here, they talk about the half cycle validation. So this means you calculate based on some formulas that are shown in there. You conduct a half cycle of 7 minutes or something like that. Yeah. And the question is then, how many bacteria are left? So this means also the pharmacopoeial chapter wants to get to know, the inspector wants to get to know your degree of overkill. How safe is the process? This is from a GMP perspective, the idea behind this half cycle validation, but it's also good for energy saving. You know, you can also get to know your process. Okay, how much is the overkill I'm applying it, and can I reduce it? Yeah, could I apply somewhat less steam here? Yes, you can, and this is the way to prove it and to validate it. It's in the pharmacopoeia. Yeah, it's accepted, it's validated. This method, just do it. Yeah, good. No more new questions. That's good. So, and here's the formulas. So I'm not going to read all that stuff out. I've marked here or underlined in blue what is really interesting. So, and here is the formulas for you, because maybe not everybody has access to the pharmacopoeias in Europe here, but yeah, I don't read all this out. This is just for your documentation. Yeah, you can look this up, and here down the formula number 3. So your validation time for the half cycle should be in between here, the log 10 n plus 1 and the log 10 n plus 3. Somewhere in between should be a time which results in the sterilizer in whatever 7 minutes, or maybe a reduced temperature or something like that. Yeah, but it's the time. It's the time that's mentioned. So that is my idea how to bring down energy consumption in autoclaves and tunnels. Yeah. To complete this topic and then move on. Yeah, not too much time left. This is the typical temperature profile in an autoclave. This is from a PQ run where you have a temperature mapping. You put these thermocouples in the autoclave. Yeah, you see them all here, colorful lines everywhere. And then there is only one different line. This is the pressure. So you have the pre-vacuum cycles here. You see you start at 2 bar absolute, and then you suck down almost to 0. So it's almost an absolute vacuum that we have here, where we are going down, and yeah, when all the others show the temperature distribution, and here once you reach the 121, the clock starts ticking. You need to wait for 15 minutes before you release the chamber, yeah, bring down the pressure and the heat again, and so this is what it looks like. And like this, you also define the cold spot. So this means the coldest, the sensor with the lowest temperature is your worst case point, and this is where you later place your monitoring sensor for the routine. Yeah, this is how it goes. Good. So much about sterilization. Last topic I've brought for you today is compressed air. Yeah. I've been doing a lot here, as said, the initial trigger to update the ISPE guide on process gases was a survey, and I'm going to show an extract on the survey on the next slide. This came from Boehringer Ingelheim, so thanks again to Stefan Neumann for this wonderful idea 5 years ago, which has really been fruitful for Merck as well, and also for ISPE. Yeah, it resulted in this wonderful new guide because we have seen that people are struggling to set a proper specification for compressed air and also clean gases like nitrogen, oxygen, CO2, whatever you may use in your facility. Yeah. And I took part in the spec chapter, and we have also written some new appendices, which I'm going to show later, with example specifications to make your life easier. Yeah? As I said, people used to define their spec based on ISO 8573, and we asked them, yeah, what is the basis for the spec? But the standard doesn't even contain recommendations for the pharma. It's a general industry standard. It just defines classes, nothing else. And so, and we saw people were struggling, and so we put down example specs, and especially on the pressure dew point, I've worked a lot. Yeah. And you're going to see afterwards what we've written down. But you could even go to a +3 pressure dew point temperature at 6 bar or so, at 6 bar operational pressure in compressed air. That would still be sufficient in most cases. It depends a bit. Yeah, you're going to see later. But then if you can't specify a +3 or higher, then you could even use a cooling dryer, which is very efficient and much cheaper than an adsorption dryer, which is typically the equipment of choice. Yeah. But we've not only looked at the spec. This is only one part. Of course, we also look at operational pressures. I see a lot of sites operating higher than 6 bar operational pressure. But why? You know, people fear that the pressure is not enough to make equipment work, to open and close valves and this and that, whatever is the application. But you know, I'm stealing a bit of their risk margin. This is what I do with all the utilities. I always look at the risk buffer, and it's quite high in many cases. And I'm trying to get this risk buffer from the people. Why 8 bar? You know, just as a rule of thumb, 1 bar makes 10% saving in your yearly consumption. That is a lot. Yeah. And another nice rule of thumb, in a compressed air, in a compressor, you are of course compressing the air as it says, and 90% of the current, the electrical power you use, go into heat. Yeah, 10% are going into really mechanical energy, meaning pressure in this case, and 90% are emitted as heat. So this is not a very efficient process, but you have no other choice. So the only thing here you can do is reclaim the heat. Yeah, reuse the heat, bring it into your HVAC system or something like that. Yeah, do something with this exhaust heat. Good. If you have non-production times, like in HVAC systems, you can lower the pressure, the base load. If you look at the base load during standby times, this is super high. This is still, you have a lot of losses during standby times. And the reason is, I can tell you where these losses happen. Of course, there's leakage testing. You all know this ultrasonic analysis. But the real reason where you lose this is the flexible tubes. You know, the SMC, Festo, John Guest, whatever type of tube, the flexible plastic tubes, these are not tight. And even if a packaging line is in standby and you don't use it, it still consumes up to 50% compared to operational mode. You still lose 50% of compressed air. It's crazy high. Yeah. Shut down compressed air and packaging lines when they are not in use. Do this automatically. It is so easy. It is just an automatic valve, and you need a signal from the packaging line. You got to look a bit when you want to restart that the packaging line doesn't go in alarm. Yeah. But it's not rocket science. Yeah. The little flexible tubes are where you lose the energy, the compressed air. Yeah. Fermentation vessels are quite hungry as well. Yeah, if you have an aerobic fermentation, that is also a big consumer of compressed air. Yeah, heat recovery, I talked about this. And here, once again, at Merck, we've already found some 2.8 million kilowatt hours of energy. So around in our 20 sites, not too bad. Yeah, so it's also really worth looking into compressed air. It's not as the saving is not as big as in HVAC systems. It is smaller, but I can tell from practice, from experience also, that it is easier to implement these actions for compressed air than for HVAC. Yeah, less work to do. So quick win. So here's the survey from Boehringer. So we had 90 companies in it, and these were all responding to monitoring, specification, and whatever topics. And this one here is the question: where does your spec come from? What is the basis for your spec? What's the rationale? And most people said here ISO 8573, but it doesn't tell you what to choose. It just defines classes. Yeah. So dew point also many people took from the European monograph, which was 10 minutes. Thanks. That specifies at 6 bar minus 27 pressure dew point temperature, which is too strict. And in most cases, we do not need to follow the monograph. Yeah. So our compressed air is not a ingredient. It's a process aid. And FDA has just issued a new guidance for industry on process gases, going even further than I do, going even beyond my opinion. Yeah. And in their opinion, nothing is an ingredient, more or less, if you look at process gases. Even for these asthma sprays, COPD sprays, as a carrier gas, FDA doesn't consider compressed air as an ingredient anymore. Good. But last but not least, before we come to the questions, I want to show you the 3 examples that we've put into the ISPE guide in Appendix 1 to 3. Yeah. Here's the FDA guidance draft. I don't know if you've access to the slides later, but I've linked it in here. So, and we've put, in this case here, we've put a +1 pressure dew point, but we also tell you the way how to assess your stuff here. And you could even maybe go to +3. Yeah, it depends. So the rationale behind is the moisture content, the relative humidity in your pipe. And we say if it's below 60% relative humidity, no yeasts and molds can grow in the pipe, no bacteria. And so this is why we say if you transfer the 60% humidity to the pressure dew point, you end up at 1 to 3. It depends a bit on your outer air temperature or the surrounding air temperature, and also your operational pressure in the system. Yeah, calculate it as written down in the ISPE guide, and then set up your dew point. For the particles and micro, we just use the clean room spec. It shouldn't be worse than the clean room. Also, this rationale can be found in ISPE guides, in the OSD baseline guide, or in the STERA guide. Yeah. Last but not least, oil. Many people used class 2 in the past. We chose this as well. It makes sense. Do not choose class 1 because the LOD of the measurements is typically higher. It's higher, so you can't measure class 1 properly. Even with a very specific Draeger tubes, it's more or less it's tough to really measure and comply with class 1. Yeah. So the first one, this was for chemicals, API sites, and OSD sites, tablets, capsules, and this and that. Yeah. And I would choose this one also for ointments and non-sterile liquids. And here the second one is biotech API. Is more or less the same. Of course, we've got a grade C clean room. And I think in Asia Pacific, the PIC-S GMP guide is quite common, including the Annex 1, and this is exactly the same classes in here. Yeah, it's been taken from the European GMP guide, Annex 1, with the classes here. Same dew point, same oil class, and yeah, I'm looking a bit at the time. Last but not least, we have two slides to go. This is the sterile slide here. The example is somewhat longer, of course. Here we have in the clean room down to class A, yeah, but we say we don't need class A conditions in our compressed air pipe. We say we put a base quality of grade C in there, and if you need a higher spec for in class B or class A because you have an outlet there, then you just apply this spec locally at this one outlet. This is pretty interesting because in water systems that would never be allowed. Yeah. In water systems, everything is stricter than in compressed air systems. Here we have the liberty, the freedom to choose a different class locally at one use point. Filters are in use, you know. the PQ is only 3 or 5 days or something instead of a year. Yeah. So compressed air systems are much easier because they are less prone to contamination. Yeah. Good. So much about the examples here. Yeah. So, and I think to also address your questions, I'm going to skip this slide here more or less. This is the calculation way how to, from the relative humidity to the pressure dew point. Yeah, I've put in a table for you here that you can just look it up. Where is your surrounding temperature, environmental temperature, and your compressed air dew point, and then you get to your humidity. That's it. So thanks a lot. I'm going now over to the questions. Here's one from Derek Chan. Based on your audit experience with EMA and FDA, how open are they to support the journey of energy saving against existing sterilization practices? Very good question. I think as this half cycle validation is in the European Pharmacopoeia, it is absolutely an accepted way and known. I mean, in the end, you must show that you have complete sterility and you should not go below these EMA decision tree conditions. This is the law, let me say. Typically, the decision tree tells you, can you use an autoclave 15 minutes for 121? If yes, do this. If not, can you use it at 8 minutes? Can you use a tunnel, dry sterilization? Can you use filtration and so on? Maybe you know the decision tree better than me. But I would say this is the absolute minimum requirement, but I see people are doing twice as much or 3 times as much as the decision tree requires. And if your validation supports this, I think they can't say anything. Yeah, good. I hope this sufficiently answered your question. And last but not least, we are coming here to the ZLD topic. So does Merck have any water circularity or ZLD projects? Has the solution been proven commercially and economically viable? Well, yeah. So of course we try here and there. I've told you about recirculating the final rinse. That's one topic we do. And I've especially one very interesting site in mind that is reusing a lot of water. This is in Brazil, our manufacturing site in Rio de Janeiro. They are reusing every little stream that they can. I don't know how high the percentage of reuse is, but they are very ambitious with this. And also, I also know other manufacturing sites in Europe that are pretty advanced here. One site in Belgium is excellent at doing this. Yeah, and if proven commercially and economically, I would say it pays off. Yeah, so I don't have numbers right now, but there is also always a reason why a site head does this. So in Brazil, they get super high wastewater fees, and there the business case is pretty easy. Yeah, it depends on the region also, of course. Good. Thank you very much. 3 minutes ahead of time. So there is still room for one more question, I guess.
So chat is still open. Thank you, Fritz. Very informative and very thought-provoking. Nothing comes up yet, but I use maybe this gap for one reference that also we wanted to mention that we have during the ISPE Singapore conference and exhibition event next week. Actually, at Friday of the event, after lunch at the Suntec Center, we have a face-to-face session planned for critical utilities community of practice. I will talk a little bit about this, and we also have here some probably news to share for the region, and also would like to inspire to get a little bit more active in exchanging topics like Fritz presented here today. Still no questions. Fritz, do you have some final words? Oh, wait, there's something.
There's one question. For a pharma site with an existing validated hot WFI loop at ATC, what would you consider the minimum evidence package needed to justify moving to a cold or lower temperature WFI loop, particularly from a microcontrol and validation perspective? Yeah, so that's pretty easy. Look up Annex 1, it tells 70. And just go down. I can tell you from a risk perspective, you can't kill the bacteria twice. If you write me a mail, you see my email, I'm gonna also show you a slide. So I remember a test that has been done on Pseudomonas aeruginosa as an objectionable organism or reference organism, where different temperatures were applied, and we looked up how long it takes to kill the bacteria. And at 70, it takes a second, 1 second. Yeah, so to kill the Pseudomonas aeruginosa. So what are you waiting for? Yeah, so good. I hope this answered your question. If not, drop me a line, write me a mail, also to the others for more questions. Talk to me. Thank you very much. And Stefan, yes, maybe sometimes I'm a bit provocative, but we need this to become sustainable. You know, it's the last planet we have. So, yeah.
I love it. No, that's what I mean with thought-provoking, because we need more energy actually in this discussion, and you very passionately brought this across. So once again, thank you for this, and thank you to all here in the audience for listening to us. And still, if there are questions, please, there is Fritz's email, or drop a note here to ISP. And maybe some of you I see next week then at the Singapore Conference and Exhibition event.
Have a great conference, everyone. Thank you so much.
Thank you. Thank you,
Have a good day.
Thank you.